Hi, my name is Ken Lasterson. I run the Microbiome prescription site, which does analysis of microbiome from multitude of providers from Nirvana Biome, Biomicide, Thrive, Vivalda, Ubiome when they were reducing it. And I'm just about to go into my second set of lessons, but before I did that, I thought it'd be good to give a very quick high-level overview of approaches. Turns out that at the moment I support 6 possible approaches, and sometimes it's probably good to stop for a moment rather than getting into an hour talk each time and having to digest everything, is to take a look over all of the dominant features between each approach. So with that said, let's get into it. So we have the overview of the lesson plan, and we can actually regroup them slightly. And apart from the introduction, we have the first 3. I'm using the term by their name shall you know them, as in by the bacteria know. The second bunch is by their fruits shall you know them. Difference between a person's name and what they do, title and actions, most people understand. And we do have the same situation which can happen with humans or with movements or concepts. We have the names and then we have the actual actions, the words and deeds. So that's basically what I'm going to talk about, is the 3 types of working with the microbiome for each one. The first one is, by name, I refer to often as the Holy Grail. There is some magical range of values which a magical ideal microbiome has, and that is what people adjust to. In other words, going for this ideal. Recent issue of Nature basically blows that whole concept out of the water. I already knew it was a bogus concept because the ranges vary by so many factors: age, diet, genetic thing. If you are dealing with ranges which are established from people which are like you, eating your same diet, in your same world location, same ethnic background, they probably work reasonably well. Here is ranges from Jason, who is in Tasmania. The ranges probably work very well for Australians and people of UK extraction who keep to sort of Australian UK style of diet, particularly Australian style of diet. And so that would be one way of doing it. However, if you are living in Mexico, you are 35% native, 35% African, and the remaining 30% European, and you're eating a Mexican style diet, your microbiome will be different, probably significantly different. So using just a set of measurements for you is probably going to be a mismatch. The second way, going in by name, is using Pubmed studies. And Pubmed studies has a similar problem. First of all, most of the studies are done on white Caucasians, which are overrepresented in the studies. Often there also are gender bias in them, and they are generally done in a specific country in the world. I know for one thing, which I did in an earlier talk, you see the shift of bacteria seen in China and seeing in a European country were opposite shifts. So we have the aspect of working from PubMed studies and finding what the shifts are, and then say, okay, this is what, because I have this medical condition and the study report such and such, this is what I need to adjust. Again, it may apply, often it will, but there are also cases where you are going down the wrong direction because the bacteria are so dependent upon so many factors, including age. The next way which we can take a look at things is by symptoms, which seems to probably be a little bit more age and other characteristic agnostic, but still having sampling bias to it. And we simply determine what patterns are seen in bacteria, stronger bacterial patterns usually than the Pubmed studies. And it gives us things where, okay, these are probably the bacterial shifts that are likely to the cause of your symptoms. And in generally, if it's high or low values, then trying to shift them towards the middle is usually a very safe activity. If you're in the middle and trying to shift up or down, there's a little bit of uncertainty of whether or not you could be doing good or doing harm. So those are the three ways of by name. You are going and you find the name of a particular bacteria and have some reason to identify it as a culprit. Now let's go over by looking at the fruits of the bacteria. Fruits means what the bacteria produces. The easiest one to consider are what's called end products, and often these products can often be independently measured by clinical lab test results. Here's an example for one of about 120 to 150 end products that are currently in the database, which is dopamine, and it goes and lists which bacteria produces dopamine. It'll come down and show you what your count is of this particular bacteria and what other people's count. But we're not interested in each individual bacteria. We are interested in grand total, the count. In other words, how many dopamine producers do you have? How many cells are producing dopamine? Once you determine you have a problem with dopamine levels, either by clinical labs or by the microbiome samples, then you need to figure out how to adjust it, which may mean having to adjust many different types of bacteria. So it's not just adjusting one, it's trying to find something to adjust a family of bacteria. Actually, more not a family, actually it's usually a collection of bacteria. Family means a specific relationship in bacteria, so let's use the term collection. So that is the end product. End products, generally the information comes from reviewing Pubmed articles, which means it's a little bit on a haphazard because you may miss an article, or a particular bacteria may not have ever been studied for whether or not it produces a certain metabolite. So there's a little bit of hit and miss there. Let's go on to the next one, which is by using enzymes. Enzymes are produced by bacteria, and we have a lovely source available on the net, not behind a paywall, called the Kyoto Encyclopedia of Genes and Genomes. And basically from there we can look at enzymes. It will give you all sorts of information about the enzymes, and from the enzymes we can go and trace back to what bacteria produces them, all the way down to strain level. Notice that it's almost always strain and species. It's not by genus, it's not by family, not by the higher level. It's going down to what is actually in an individual species or strain of bacteria, where the genes are going to be far more stable and less subject to wild variations. When you go up to the genus or the family level, you get a lot more variations in the genes. So just gives you the list of things so you can go and see whether or not you have just enzymes abnormally high or low, and if you have high or low, it will give you some hints as to which bacteria are the ones which produce it in either excess or in insufficient numbers, and then we can try figuring out how to adjust the collection of producers to produce more or produce less. The next one in the list is a keg modular, which is somewhat similar. It is a different way of seeing how the bacteria interact and function with each other. And again, we can from them identify the bacteria involved, and actually it's very nice because if you go to the site, which is linked to from the web pages, you will see a lovely thing called taxonomy here, and if you click the taxonomy, you will see the bacteria involved, and you can start working from there, saying, okay, we have too much or too little of the bacteria involved in that. Here is a example, and let's just sum up the methodology. End products are done by searching Pubmed. It tends to be ad hoc, it tends to be incomplete, but it tends to be familiar. Most of the names of the end products, like dopamine, are things people recognize, so it has a familiarity to it, which means that people could feel more comfortable and less in the dark, even though they may have no idea of everything. They feel comfortable with the name dopamine. Cake enzymes comes from the enzymes tracing back to the gene to produce it, from the genes we trace back to the bacteria. So it is a multiple step trace back. Dataset is open. Every lab could offer this feature because the data is there. They know the bacteria at the species or strain level, so they could go and literally do a cake enzymes report with all the other reports that they offer you. The cake modular, as I said, is a nice because it's a one click. For example, here I clicked the arginine biosynthesis, and particularly how one chemical gets changed to another chemical, and clicking on that I get a list of the organisms that does it, and you can see there's a whole bunch of different bacteria, and we name the specific ones, and the specific ones are the ones that have been studied. The nice thing about the KEGG Encyclopedia, the Kyoto Encyclopedia of Gene and Genomics, is it basically pre-assembles all scientific literature. It's a massive undertaking which has been going on for years, and it's just a suite. It's open if you are wanting to access it in a data format. You can do an FTP download. For some type of organization, they will charge you for having the FTP access, so you need to check with them. But again, what we have is we have free methods which are by fruit, which is, to me personally, the preferred way of doing it. One is by searching Pubmed. This is an open data source, but it's time consuming to search. We have the cake enzymes, which comes right from the Kyoto site, which is sweet. So it's just a matter of looking it up and tracing the data through, which can be done by software if need be, or by downloading the FTP of the databases and just looking up in their database. And similarly to cake modular, which is the same thing. So we have three ways which we can evaluate your bacterial mixture by what fruit it bears, what chemicals they produce, what metabolites they produce, etc., which makes it sweet and probably the preferred way of going, because generally the level of certain enzymes is what the body needs around the world, so it is likely less biased towards particular DNA or to a particular diet. What we have also is bacterial substitution. You don't have any of one bacteria, but you have a bunch of another bacteria which happens to do the same function, produces the same fruit, or in some combination, some substitution, it produces the same fruit, which means you shouldn't be too concerned about which bacteria you have. You should be concerned about what fruit the bacteria produces. Enzymes, end products, the cake modulars. So, best method conceptually is by fruit. It identifies defects in existing metabolisms, and more importantly, why? Because it identifies which collection of bacteria are involved. In other words, a lot less making wild guesses or speculations in the dark. Methods can be done by all the labs. There's nothing proprietary for it. The data sources are open, easy to do. It's just a matter of the people at the labs sitting down, getting the data imports, being able to process and understand the data, and giving you reports. The challenge with this method is we're not dealing with a single bacteria or even a small set. of bacteria. We're dealing with a collection of bacteria which may go across many different families, genus, or even orders. That means trying to adjust it becomes more complicated because you don't have a simplistic one-on-one adjustment you're trying to do. You're trying to change a suite of items and compensate appropriately. My general impression is to do analysis of what you should do to correct it actually requires artificial intelligence technique because of the number of items involved, the complexities, and whatever you do is going to have side effects elsewhere. So it becomes an interesting problem. My academic background is operations research, and it's an interesting problem. I like it. However, not everybody has the same propensity, and it is also a very technical problem. Okay, with that, I think we are done. And so this was a quick overview of 6 ways of trying to figure out how you need to correct or could correct your microbiome. And it's up to you which ways you want to try. I have my own preferences. And I tried actually on my side allowing people to do it by however they want. Even though I have a preference, I believe it's a choice people should make for themselves. Okay, that's it.